Okay guys, so in this video we're going to talk about the biosynthesis of tyrosine. Like tyrosine is a very important molecule in biosynthesis pathways, especially in shikimate pathway. One of the major metabolites in our shikimate pathway is tyrosine. This is a very quick one, not that much amount of steps in here. Let's go through. I hope you remember our prephenate anion. Like if I go back to our previous video, like we have discussed this about our previous video, right? So here we have talked about how the prephenate anion is formed. So I'm going to go forward with our prephenate anion. You can go with the neutral compound too, right? So I'm discussing the synthesis pathway, or otherwise the mechanism with anion. There's the same thing with the neutral compound too, just the hydrogen, right? So it's just a neutral compound, both are the same. Okay, so the first step would be NAD plus. I hope you remember like this NAD plus, NADH, these functions, right? These are this NAD plus and NADH. These are the components that can exchange hydrogens. NAD plus means it lacks a hydrogen, so it can take a hydrogen from a particular molecule. So here you can see this is a OH group there. So this hydroxyl group has converted into a carbonyl group. The hydrogen is being removed by this NAD plus. With the reaction is moving forward, this NAD plus is taking a hydrogen and converting to NADH. So this hydroxyl group converted into carbonyl group. And the next step would be removal of the carbon dioxide. So I have talked this mechanism also in our previous video. So let me explain again if you want to clarify it. So here I have drawn a molecule. You have here carbon, oxygen and anion. Okay, now what happens is like this valence electron will form a double bond here. So this carbon will make itself unstable. So this bond will be broken. So you can see now this carbon is unstable. So here this is rearranging into this position. Now this carbon is again unstable. This will attack into a proton to get stable. So now here the other part is the same. Other part is nothing changes, same thing. So you can see like carbon, oxygen, this is leaving from our main molecule, which means a carbon dioxide is leaving. And you can see this carbonyl group is converted back into hydroxyl group, but the aromatic ring is formed in the middle. So this step is like really fast. Like the aromatic ring is very stable. Like if some molecule contain an aromatic ring, which means it's stable. Like this is not a this aromatic ring formation. It's a very stable process. Okay, so you can see now we have this particular molecule. Now again, I hope you remember this transamination process. Transamination process, which we have discussed in our previous video also. What happens? This is the glutamate. This is the glutamate molecule. Here you have an amine group. This amine group and this carbonyl group will be exchanged. You can have this tyrosine molecule, and plus the other product is this glutamate, but not with the amine group. Instead, it contains a carbonyl group. This transamination is basically the exchanging of this oxygen and NH3 plus. So NH2, if you go with the neutral compound, so I am using anions, that's why I have put a NH3 plus. So this is a very quick and very small biosynthesis pathway, easy to remember, easy to memorize, like nothing a big thing, nothing so much complicated steps are happening in this particular reaction, but things are like you need to understand these kind of steps very quickly. Not quickly, I mean like you have to understand this one. I hope most of the papers you get, like question won't be asking like write the mechanism for the tyrosine biosynthesis. Not that like questions. Most of the papers contain the question like filling the blanks kind of thing, right? the mechanism is given, but instead of this molecule, it will put a blank here. So you need to draw the molecule for that particular thing, and the reagent will be, or otherwise the steps would be blank. So in here this will be blank and the name will be blank. So you need to identify what is the molecule here and what is the process, the transamination, that kind of thing. So these are the kind of question I think you would get. Like, it depends based on the university which you are referring and the college you are learning, but the basics is you need to understand each steps, like especially what are the function of this NAD plus NADH. Like if you write the NADH on here, it's completely wrong, right? So these are the kind of things you need to be very focused on. I hope you got the idea for this particular reaction. Let's move into our next video.